Today we shall be discussing the translocation of photoassimilates. Phloem transports the organic products of photosynthesis throughout the plant, that is, from mature leaves to areas of growth and storage, including the roots, via a process called translocation. The products of photosynthesis are also called photoassimilates, and by translocation, we mean the long-distance transport of photoassimilates. That food is translocated by the phloem is indicated by several evidences. One is girdling. In girdling, the phloem tissue is removed by separating the phloem, that is the bark, from the xylem or the wood at the vascular cambium. The girdle interrupts the downward flow of food. Eventually, the plant dies because the root system will starve from lack of nutrients. An analysis of phloem exudate provides more direct evidence in support of the conclusion that photoassimilates are translocated through the phloem. Unfortunately, phloem tissue does not lend itself to analysis as easily as xylem tissue does. This is because the translocating elements or the sieve elements in the phloem are, unlike xylem vessels and trocades, living cells when functional. These cells contain cytoplasm with slime bodies or P-bodies with an inherent sealing action when cut. The slime plugs aggregate at the sieve plate found at the end walls to seal the wound, followed by wound callus. Thus, the sieve tube elements do not exude their contents, making collection and subsequent analysis difficult. One solution to the problem of obtaining the contents of sieve tubes uncontaminated by other cells was provided by insect physiologists studying the nutrition of aphids. Aphids are one of several groups of small insects that feed on plants by directly inserting along mouthpart, the stylus, directly into individual sieve tube members. When feeding, aphids are anesthetized with a stream of carbon dioxide, and the stylus is carefully severed with a razor blade. The phloem sap continues to exude from the cut stylus for several days. Exudate from severed stylets provides a fairly accurate picture of the composition of the phloem sap. A third evidence for phloem translocation of photoassimilates is provided by use of radioactive tracers. Attached leaves were allowed to photosynthesize in a closed chamber containing radiolabeled carbon dioxide or carbon-14. After 10 minutes, the radiolabeled photoassimilate being transported out of the leaf was immobilized by freezing the petiole in liquid nitrogen. Cross-section of the frozen petiole was prepared and placed in contact with an X-ray film. The resulting image on the X-ray film indicated that photoassimilate being translocated out of the leaf was localized exclusively in the phloem. Results from analysis of collected sap indicate that the translocated carbohydrates are all non-reducing sugars. Most researchers believe that the non-reducing sugars, like sucrose, are the principal compounds translocated in the phloem because they are less reactive than their reducing counterparts. Thus, reducing sugars like glucose, mannose, and fructose are generally not translocated in the phloem. Some families, like the Cucurbitaceae family, translocate in addition to sucrose, the raffinose series, that is, sucrose bound to varying numbers of galactose molecules. Raffinose consists of sucrose bound to one galactose molecule. Stachyose consists of sucrose bound to two galactose molecules, and brevascose consists of sucrose and three galactose molecules. Others translocate their photoassimilates as sugar alcohols like mannitol or sorbitol. Inorganic ions are cytoplasmic constituents of translocating cells and do not necessarily represent translocated photoassimilates. Plant hormones like auxin, cytokinin, gibberellins, and abscisic acid were also detected in the phloem exudate at low concentrations. Proteins found are enzymes and P proteins. Glutamic acid, an amino acid, and glutamine, its amide, are important tritogenous compounds in the phloem, in addition to aspartate and asparagine. The direction of long-distance translocation in the phloem is from the source to the sink. By source, we mean a producer of photoassimilate like a mature leaf, or a net exporter of photoassimilate like the bulky root of kamote or carrot. By sink, we mean a net importer or consumer of photoassimilate, or a storer of sugar like a growing fruit. A storage organ such as the tuber may be either a source or a sink. When the storage organ is still stockpiling carbohydrates, it is a sugar sink. The storage organ becomes a source as its starch is broken down to sugar, which is carried away in the phloem to the growing buds. Assimilates are partitioned among sinks, which are competitive. Partitioning depends on three factors, one of which is the nature of the vascular connections between source and sinks. Photoassimilate will move preferentially toward the sink leaf above and in line with the source leaf. In this figure, the source leaf is leaf 14, which was applied for 4 hours with radiolabeled carbon dioxide. 1 week after, the 14 carbon label was translocated mainly to the sink leaves directly above the source leaf, that is, leaf 1 and leaf 6, as indicated by the degree of radioactive labeling of these leaves. Partitioning of assimilate among sinks also depends on the proximity of the sink to the source. The upper mature leaves provide photosynthesis to young immature leaves and the shoot tip. The lower leaves supply photosynthesis predominantly to the root system. Intermediate leaves, on the other hand, export photosynthesis in both directions, that is, towards the tip and towards the base. Partitioning of assimilate among sinks also depends upon sink strength. Sink strength is a measure of the capacity of a sink to accumulate metabolites. What determines sink strength, however, is not known. In this experiment, the supply of photoassimilates was altered by reducing the supply of carbon dioxide from high to low, and the dry weight increase of various parts was monitored over the grain filling period. Reducing photoassimilate supply had virtually no effect on the grain weight, which means that a higher proportion of the carbon was translocated to the grain even when carbon dioxide level was reduced. The difference was made up by the equivalent decrease in the proportion of carbon directed to the roots. Roots and the developing grain are competing sinks. When the supply of photoassimilate is limited, it is preferentially directed toward the sink with greater strength. in this case, the grain. In plants, the developing fruits are the major sink, and crop productivity and quality depend on an appropriate source-sink relationship. Limiting the number of fruits per plant often improves fruit size, that is, the fruits are bigger, the flesh are thicker, and the fruits are also sweeter because there are fewer sinks to partition the assimilates. You simply break the fruit off with your fingers to thin fruit trees. No special tool is required. The mechanism of translocation in the phloem is by pressure flow or mass flow. That the sieve element is under pressure is shown by the honeydew droplet which comes out of the anus of the apid as the tube force feeds the insect. Honeydew droplet consists of phloem sap minus some of the nutrients absorbed by the insect. That the sieve element is under pressure is also shown by the prolonged exudation of phloem sap from excised apid stylets. The total volume of exudate may exceed the volume of an individual sieve tube by several thousands. The mechanism of phloem transport is bidirectional, that is, from mature leaves upwards to areas of growth or from mature leaves downwards to areas of storage. Bidirectional transport in adjacent sieve tube elements can occur. However, simultaneous bidirectional transport in a single sieve tube element has never been demonstrated. Shown here is a diagram of pressure flow. The loading of sugar into the sieve tube element adjacent to a source cell reduces the water potential inside the sieve tube member and causes osmotic uptake of water from nearby xylem elements. The uptake of water increases the hydrostatic pressure or the turgor pressure in the sieve tube element. The pressure is lowered at the sink end when sugar is unloaded into the receiver cell. Removal of sugar at the sink increases the water potential of the sieve tube element and causes water to flow out of the tube and for water to return to the xylem. This pressure differential, that is higher pressure at the source region of the sieve tube element and lower at the sink, causes a flow of water from the source to the sink, with sugar being carried along passively. The phloem flows from the source to the sink at rates as great as 1 meter per hour, faster than can be accounted for by either diffusion or cytoplasmic streaming. The initial transfer of assimilate from the mesophyll cells into the sieve tube elements at the source end is called phloem loading. The movement of sucrose from the mesophyll to the phloem parenchyma is by the symplast through diffusion. From phloem parenchyma, there are two possible routes for sucrose. One is it may continue to the companion cell and the sieve tube member complex via the symplast. If sugars freely diffuse from the mesophyll into the companion cell sieve tube element complex, they should be equally free to diffuse back into the mesophyll. How then is it possible for the sieve tube element companion cell complex to accumulate sugars by simple diffusion through the plasmodesmata? Based on studies of phloem loading in cucurbita species, a polymer trap model has been proposed to account for symplastic loading. According to the polymer trap model, sucrose diffuses from the mesophyll into the bundle sheath cell and into the companion cell, called the intermediary cell in this illustration, through the connecting plasmodesmata. In the companion cell, the sucrose is converted to an oligosaccharide such as raffinose, which is too large to diffuse back through the plasmodesmata. The polymer, that is raffinose, is thus trapped in the sieve tube element companion cell complex and rises to be carried away by mass flow. Sucrose can continue to diffuse into the intermediary cell or the companion cell because its synthesis in the mesophyll and its utilization in the companion cell or intermediary cell maintain the concentration gradient. From the phloem parenchyma, another possible route for sucrose to reach the sieve tube member is by a combination of apoplastic and symplastic pathways. From the phloem parenchyma, sugars first pass into the leaf apoplast. The sugar is subsequently taken up either in the companion cell or in the sieve tube member by sugar hydrogen ion symport to be translocated out of the source region. Proton pumps at the plasma membrane first generates a hydrogen ion gradient which drives sucrose uptake across the membrane via a cotransport protein that couples sucrose transport with the diffusion of hydrogen ions back into the cell. Evidence indicates that the sucrose hydrogen ion cotransport mediates sucrose transport between the apoplast and the symplast. The sugars are actively loaded into the companion cells and the sieve tube elements from the apoplast. Sugars loaded into the companion cells are taught to move through the plasmodesmata into the sieve elements. For those plants exhibiting the symplastic pathway of phloem loading, the sucrose hydrogen ion cotransport may exist as a mechanism for retrieving sucrose that leak from the source cell or from the sieve element to prevent unnecessary loss of sugar from the transport stream. It has been observed that those plants with companion cell sieve tube element complexes were symplastically isolated exhibited characteristics of apoplastic loading, while those with abundant symplastic connections exhibited characteristics of symplastic loading. By phloem unloading, we mean the final transfer of assimilate of the sieve elements into the target cells. There are two mechanisms of phloem unloading. We have by symplastic pathway or by apoplastic pathway. In the symplastic pathway, the sucrose flows via the interconnecting plasmodesmata down a concentration gradient from the sieve element companion cell complex to sites of metabolism in the sink. The gradient and consequently the flow into the sink is maintained by hydrolyzing sucrose into fructose and glucose. Another mechanism of phloem unloading is by apoplastic pathway, which could involve passive unloading into the apoplast, as shown in maize seeds, or active unloading into the apoplast, as shown in the legume seeds. In passive unloading, once in the apoplast, sucrose is hydrolyzed by the enzyme acid invertase, forming glucose and fructose. This reaction is essentially irreversible, and the hydrolysis products, glucose and fructose, are actively taken up by the zinc cell. Once in the cell, they are again combined as sucrose and actively transported into the vacuole for storage. Hydrolysis of sucrose in the apoplast, perhaps combined with the irreversibility of the acid invertase reaction, serves to maintain the gradient and allows the unloading to continue. This pathway seems to be prominent in seeds of maize, sorghum, and pearl millet. The third pathway for phloem unloading indicates that, at least in legumes, sucrose is unloaded into the apoplast by an energy-dependent carrier. The nature of the carrier has not been conclusively identified, but evidence to date suggests it is probably the same sucrose hydrogen ion cotransporter described earlier. As with phloem loading, there does not appear to be a universal path for phloem unloading into the developing embryo.